Hello, and welcome to the Active Motif webinar series. My name is Mason Brooks, and in this episode, we'll be getting some updates from my colleague and Active Motif R&D scientist, Dr. Joshua Messenger, about how he's been able to apply the use of automated sonication equipment to increase the throughput and really enable the epigenetic analysis and study of clinical samples in ways that just weren't possible before. So here's Dr. Messenger to tell you a little bit more.
Hey, Mason. Thank you so much for having me back. I'm really excited to be back to talk about my work here at Active Motif. Today's talk entitled Bridging ChIP-Seq and Translational Research with PIXUL High Throughput Sonication. So today we're going to talk a lot about our new PIXUL Multiwell Sonicator and how it's helping us process clinical volumes of samples to facilitate translational science and bench to bedside research. So I think we all know out there that epigenetics has huge potential for clinical medicine. Many of us spend more hours in a day than we can even understand thinking about epigenomics and how we can take what we're learning at the bench and really help a patient population and get what we're learning through our in vitro assays to the bedside. So I just want to highlight a couple of things that are on this slide here. We know that gene transcription and gene expression can be regulated by DNA methylation, which are all facilitated by enzymes called DNA methyltransferases. And we have a number of small molecules that can regulate the activity of these methyltransferases. Histone modifications themselves can also greatly regulate gene expression, which is facilitated by histone acetyltransferases and histone deacetyl enzymes, HATs and HDACs, and we have small molecules to regulate all of those enzymes. And if we could actually target the expression of these enzymes, where maybe we have a gene that's on in a disease state that should be off, or a gene that's off in a disease state that should be on, and we can target these regulators of epigenomics to either shut off or turn on these genes, we could have much more targeted therapeutics. We can make a much bigger impact in the clinical space with what we know about epigenomics. However, despite everything we've learned over the years about epigenomics and where all these histone modifications are located and how they regulate gene expression, the clinical implementation of that epigenetic data has been really slow. And there's wide availability of these epigenomics data. I think many of us are familiar with the ENCODE project and the incredible work the ENCODE consortium does to detail where these histone modifications are in the genome across multiple cells and tissue types. But despite how much we've learned through these experiments, actual implementation of this has been very slow in the clinic. And there are unfortunately a number of reasons why. First reason is that there's often limited clinical samples. I think we see this a lot as researchers, that you really want to study this disease state, but it's just hard to get your hands on enough tissue, or maybe there just isn't enough tissue. And the tissue that you can get your hands on is very precious. And when you process that for epigenetic assays, you often find there's not enough material to answer relevant questions. So if you process your tissue and you make chromatin and do your input prep and you quantify the amount of DNA there and you come back with less than a microgram of DNA, unfortunately, you can't do a ChIP experiment. And then the sample isn't as useful for asking that question to understand the disease state. It's often very linear and cumbersome to make chromatin. I think a number of scientists would agree that chromatin preps are not the easiest thing in the world. Probe tip sonicators can often do one sample at a time. So that often dictates how many samples you can process in a day. And you would need an army of epigenetic scientists constantly shearing chromatin to have enough material to do these epigenetic assays. And despite that, even if you were doing that, oftentimes you get inconsistent shearing across your samples, which means that the DNA is not appropriately sized after sonication, and it's not suitable for ChIP-Seq. And that can really be frustrating because that means that a lot of samples that you have end up getting thrown away through no fault of your own, but just because of inconsistencies in the actual processing methodologies. And lastly, I just want to highlight that there are often barriers in tissue homogenization. So we are very intact organisms. We don't like to come apart, which is really great for maintaining our health. But when we're trying to study the epigenome, it's almost the exact opposite of what we would like. We need these tissues to come apart and make single cell suspensions. And if they don't make single cell suspensions, Then there's low amounts of DNA that's actually purified during chromatin input preps. And we circle back on the first point where there's not enough material to answer these relevant questions. So these are many of the problems that epigenomic scientists are facing on a daily basis, and some of the things that we here at Active Motif are trying to overcome to allow epigenomics to really make an impact at the clinical interface. So how do we bring epigenetics from the bench to the bedside? We need a high throughput and consistent method for processing chromatin from mammalian tissues for epigenetic studies. We need to know that we can process tissues from different clinical disease states, whether they be from human or mouse or any other model system that we might be looking at, that we can process them with high throughput, consistently and reliably, and that the material that we're going to come out with on the other side is of high enough concentration and quality that we can go into these ChIP-Seq or other epigenomic experiments. Well, our answer for that is the PIXUL Multiwell Sonicator, and that's something that we launched in 2020 that we've been very excited about here at Active Motif. This is a 96-well plate-based Sonicator. It is capable of consistent high throughput shearing of chromatin. And you can do multiple parameters in one run. What I think is really, really cool about the PIXUL Sonicator is that each column in the 96-well plate is controlled by its own set of transducers. So as a result, you can run up to 12 different optimization protocols at one time. Each column can run its own parameters. So as a result, if you're working with a tissue that you're unfamiliar with and you have to run some optimization on its sonication, you could do all of that on one plate in the same run, making optimization experiments really, really easy. It also means that you could sonicate tissues at different parameters on the same plate if you needed to. It's very inexpensive consumables. These work with traditional U-bottom 96-well plates that are often found in most standard laboratories. So we're really excited that you don't need 100 samples to justify running PIXUL because it's inexpensive to run, but you can run 100 samples if you have that volume, which we think is a really nice option. And this allows for the sonication of clinically relevant numbers of samples all at one time. We know from seeing clinical studies and from performing these high complex experiments that you need a large number of samples to generate enough data points to actually draw conclusions about what you're seeing in the epigenome and whether or not that can actually be something that helps in the clinical space. And we need a lot of tissues and a lot of samples to be able to actually draw those conclusions. And now with the PIXUL Sonicator, being able to do 96 samples at one time, we can actually process at a speed that is high enough to suit the volume of samples that we will need to study for these clinical assays. So far, we have tested a number of different tissues across human and mouse on our PIXUL Sonicator to understand how well it sonicates chromatin for downstream epigenomic assays. We've looked at 10 human tissues so far: kidney, liver, uterus, colon, adipose, ovary, PBMCs, heart, brain, and lung. So a real survey of tissues that you can get from the human body, and 8 different mouse tissues with the kidney, liver, spleen, heart, lung, brain, colon, muscle. We found that human tissues could be sonicated on PIXUL in 72 minutes under a standard set of parameters, and I'll show some of that data in a second, and that mouse chromatin could be sonicated to full shearing in about 36 minutes. So we did find a little bit of species differences, but like I said before, you could run different parameters on the same plate if you needed to. So this is not a barrier to throughput. So what we're looking at here is this is sonicated chromatin purified from an input prep done on human brain. This is fragment analyzer data where we can see in the unsonicated control lane, which is the first lane, that there's really no migration of low molecular weight material as expected. This should be mostly intact genomic DNA. But when we put this sample through PIXUL and we sonicate for 72 minutes on PIXUL, we found that there's a nice enrichment of material around 300 base pairs, which is the ideal size for ChIPSeq. We did the same experiment on mouse brain. Now we're looking at tape station data, so the scale is a little bit different. But what we can see in the first lane where we do not sonicate with PIXUL, which is the PIXUL N lane, there's a retention of high molecular weight material, which would be considered genomic DNA, but no migration of small molecular weight sonicated material. But you see when we put this material through PIXUL and we sonicate for 36 minutes on PIXUL, we get a nice enrichment at 300 base pairs, similar to what we see in the human brain as well. And when we quantify the amount of sonicated material, what we can see is that when we don't use PIXUL Sonication, there's really no sheared material as expected. It should just be an intact genome. But when we sonicate with PIXUL and we're able to break that down, we find that in human, about 80% of material, and in mouse, about 75, 72% of material is fully sonicated, meaning that it's between 100 and 900 base pairs. I'm highlighting here the QC metric of 70% with a dotted line. This is our internal control for material that we feel is appropriate to go into ChIP-Seq. And what we can see is that for both of these samples, we've surpassed our 70% metric, which is really wonderful and means that this chromatin is suitable for downstream epigenomic assays. If we look at all the tissues that we sonicated on PIXUL, these are human tissues, what we're able to see is that most, if not all of the tissues surpass our 70% QC metric. Kidney, adipose tissue actually nearly sonicates to 100%, which is really wonderful. Some tissues are a little bit tougher, like heart. You can see that heart is approaching that 70% mark, but it's just a little bit under it. But in data that I'll show later, this material still performed very well in ChIPSeq. But for the most part, this standard set of parameters at 72 minutes on PIXUL is enough to sonicate all chromatin from all of these different sources to 300 base pairs in a way that surpasses our 70% QC metric. We see the same trend in our mouse PIXUL chromatin, where most of the tissue sources chromatin are surpassing our 70% QC metric. Again, this is with half the amount of time for human. This is 36 minutes. We see that heart and colon are a little bit below our 70% QC metric, but I'll show later that this chromatin did perform very well in ChIPSeq. And what we're excited about is that this means that for each of these organisms, we can sonicate chromatin under a standard set of parameters and come out with material on the other side that is appropriately sheared, of the correct size, and suitable for downstream ChIP-Seq application. So this is really exciting because we feel that this throughput is high enough to accommodate clinical volumes of samples. So next, now that we had this chromatin that was suitable for ChIP-Seq, we performed ChIP-Seq and made some libraries. So here we're looking at NGS libraries that we prepared from human brain. So the input library is seen here with enrichment at about 400 base pairs, and we perform ChIP for H3K9 acetyl and make an NGS library with the resulting purified DNA. We get enrichment of also about 400 base pairs, which makes sense as this represents ligated material during NGS library prep from the 300 base pairs that was enriched during sonication. And we see the exact same profile when we look at mouse brain. The input again enriches at about 400 to 500 base pairs, and we see a really strong library from H3K9 acetyl ChIP DNA, which we performed in mouse brain. So we did this in all the tissues that I mentioned previously. We prepared H3K9 acetyl and input libraries and sequenced them with paired end reads. And this is just what some of that data looks like. So now I have all of the tissues that we tested back together on the same browser track. This is all H3K9 acetyl ChIPSeq data. And you can see with each color set represents a different tissue, and that its ChIPSeq track is presented over its input, which is the background that we're calling peaks over. And right now we're looking at the RNA polymerase 2A subunit gene, or the POL-R2A locus, which is a standard housekeeping gene that should be expressed in all cells, as RNA polymerase 2 is necessary for gene transcription and making your cells do what they do. So what we can see is there's really nice H3K9 acetylation at the promoter, as we would expect in all of these different tissues at the POL-R2A promoter, which is really great, as we would expect RNA polymerase 2 to be active and expressed in these cells and as necessary for transcription. But when we switch our attention to a different gene locus, like the CD3D locus, which specifically marks T cells, as this is a subunit of CD3, we really only see strong H3K9 acetylation here throughout the entire gene body in PBMCs. And this makes sense, as PBMCs, or peripheral blood mononuclear cells, are enriched for immune cells, which T cells are, and CD3 is a marker of T cells. And we don't see much crosstalk of the CD3 H3K9 acetylation in any of the other tissues that we surveyed, which means that we're getting really strong tissue specificity data from these experiments using PIXUL chromatin, which is really exciting for us. We then did the same thing in mouse. Here are all the mouse tissues that we tested. what you can see again is there's really nice H3K9 acetylation at the promoter for RNA Pol2, which is great as we expect. And same as before, RNA polymerase 2 is necessary for transcribing genes in cells, so we expect this to be on. However, when we shift our attention to another gene locus, such as ADD2, ADD2 is a gene that's been shown to be a tissue-specific marker for mice in brains. And what we see is there's really nice H3K9 acetylation at the promoter of ADD2 in the brain, but we don't see this in any of the other tissues that we surveyed, which again is really exciting and adds some integrity to our data, that not only are we getting really clean data, but it's really high quality in that we can pull out these tissue-specific differences between all of our different samples. So I just want to come back to our original goal. The goal here is to bridge the bench and the bedside and to get as close as we can to making translational and precise therapeutics as we can. PIXUL represents a solution for us to do that. It allows for high throughput sonication of primary mammalian tissues under a uniform set of parameters. And all of this chromatin performs reliably in ChIP-Seq. And this is really exciting because, like I said before, it means that we can process the number of samples that we need fast enough and reliably enough for downstream epigenomic assays. And so something I didn't mention, but I do want to draw attention to, is that epigenetic profiling via ChIP-Seq in 10 human tissues and 7 mouse tissues, 3 histone marks, and a transcription factor was done by a single scientist in a few months. So today I showed H3K9 acetyl data for all these different tissues. I also have H3K27 acetyl data, H3K36 trimethyl, and CTCF. And this was all done by myself over the course of about 4 months with optimization. With the parallel processing I was able to do, these experiments happened very quickly, and I was able to generate a lot of data across all these tissues in both organisms very quickly. Previously, this is something that would have to be done linearly, and as a result of that would take a lot longer, probably about 6 months longer to get all of this data collected. And we know through watching disease states that time is of the essence. So being able to process these things faster and in higher volumes means that we can make a difference faster in the clinic and really touch on personalized and targeted therapeutics. And as I previously stated, it's impossible due to the previously linear nature of processing samples. And that PIXUL represents a new robust technology for consistent and fast chromatin shearing for clinical volumes of samples for epigenetic studies. And this, I think, is how we're going to get epigenetics to the translational interface, is through these high throughput processing of samples that we're now able to do with our PIXUL Multiwell Sonicator. So I want to come back to our little diagram here about why epigenomics has been so hard to put into the clinic. We mentioned the limited clinical samples, the linear and cumbersome nature of making chromatin, the barriers in tissue homogenization. Well, PIXUL doesn't solve everything, but it does solve a lot. It does help to overcome the linear nature of making chromatin, and it also helps to overcome the barriers in tissue homogenization, where we found that PIXUL sonication is robust enough to even break up small chunks of tissue and really get DNA out of these single cell suspensions that are being put into these plates. So we really think that PIXUL offers a unique solution to enabling clinical levels of epigenomics, and we're excited to see what kind of personalized therapeutics come out of the epigenomics world in the future, hopefully with PIXUL right alongside of it. So with that, I just want to thank everyone for coming out and listening today and learning about the exciting work that we've been doing here at Active Motif. If you want any additional information, please feel free to visit www.activemotif.com/PIXUL or reach out to us at tech_service@activemotif.com if you have any further questions about any of the technology or methodologies presented today in this presentation. Thank you so much for everything, and I hope to see you again next time.